Hi, welcome back to the last webinar of this second COVID-19 series. I hope you've enjoyed them so far. This is Ring again from the Congress Director of the World Vaccine Congresses. Before we begin, I just want to thank our webinar sponsor, TriLink Biotechnologies, for supporting us to help deliver today's webinar with Moderna, as well as our first series that took place last month. If this is your first time with us, please be aware that you can submit questions using the control panel throughout the webinar, which I will collect. But the Q&As will only begin after the presentation is finished. And this session is also video recorded, and you'll get a link to it in 24 hours over an email link. So let us begin. So today's final presentation is delivered to us by Moderna, as you know, who's done a tremendous amount of work towards developing a COVID-19 vaccine. So I would like to keep introductions fairly short today. So I will now pass over to our much anticipated wonderful moderator, Dr. Krause, Deputy Director, OVRR CBER at the FDA, to properly introduce Dr. Zaks and his session. That's it from me. Over to you, Phil. Thank you.
Yeah, welcome everybody. So Dr. Zaks is the Chief Medical Officer at Moderna. As you know, Moderna is a company that has a platform technology that brings RNA into cells and allows the expression of proteins from those RNAs. And one of the possible outcomes of that protein expression, of course, is immune response and vaccination. And Moderna has been right at the center of developing COVID-19 vaccines, and in fact, they're in collaboration with NIAID. Their vaccine was the first one to enter clinical trials here in the U.S. Tal has a lot of industry experience, actually much of it in oncology, but he's been right in the thick of things at Moderna as well, and also has an academic appointment at the University of Pennsylvania. His talk will be entitled Moderna's Vaccine and Trial Efforts Against COVID-19. So welcome, Tal, and we look forward to your talk.
Thank you, Phil. I apologize to the audience. I think this webinar will be a little bit shorter than the usual. I think we're going to close it by around 11:40. My talk is going to be relatively at a high level, but I do want to give you a sense of why we believe this technology makes sense for this application to prevent COVID-19 and where we are in the journey. So this is the schematic of what we're trying to do. It's the same schematic that really underlies everything we do at Moderna, which is if we can get mRNA into a cell and convince the ribosomes that that mRNA is kosher and they should make protein out of it, then we can get the protein made in the way that it would be made in normal biology. And so if you can actually overcome the delivery piece, then and you got the right chemistry and everything else and process to make the mRNA, we've learned over the years that indeed this works as advertised, in the sense that we get the proteins made and the ribosome actually can't make a wrong protein. The beauty here is that you're using the body's own cellular machinery to make protein. Now, this notion of mRNA to protein, that's actually sort of the central dogma of biology, and all life works that way. That's the underpinning of our technology. And so the beauty for vaccines is sort of obvious. You actually need a relatively low amount of protein when you compare to therapeutics. The delivery mechanism is a needle and a syringe, and I'll come back to that in a minute, but something we're all very familiar with. And the biology is the correct one, in the sense that when you get an mRNA that translates a protein from within the cell, you actually, without intending it, because you're skating down with evolution here, you're getting T-cell responses as well as antibody responses. And in every application where we've looked, we've seen a nice T-cell response. And in fact, the T-cell response component is the underpinning of a totally different application, that of cancer vaccines. Now, I spoke about the needle here. What really happens is that we inject in the LNP-IM into a muscle. The action is at the local lymph node. We've shown this in non-clinical models quite nicely. The LNP ends up discharging its mRNA mostly in antigen-presenting cells that, you know, they think it's a virus as far as they're concerned, so they start making protein, but they are then educating the rest of the immune system, and you end up getting B cells as well as T cells. The level of T cell activation, even though T cells are always tricky to measure, and I say this as somebody who most of his life as an immunologist spent on the T cell side of the equation. But if you look at the strength of the antibody response, the slow decay, what you get out of a boost, you know that those are happening because you've got T cell help, even if you haven't demonstrated it directly. This is a very flexible platform, and so it allows us to do a whole bunch of things. And over the course of our history in infectious disease, we've actually put our first product in the clinic as recently as the end of 2015. So not a long time, but in that time, we've done a whole bunch of stuff. We've done relatively simple antigens. You can see here in the case of HMPV and PIV3, two respiratory viruses. The beauty is that you've got two antigens for two different viruses going in the same LNP formulation. And we've shown in phase 1 trials that that allows us to actually immunize against both viruses independently. This is, of course, thinking ahead to the ability to combine it even with RSV for a pediatric respiratory vaccine. We've done more complicated antigens that I'll come back in a minute, but the ability to do complex and combinations of mRNA is sort of one of the beauties of this platform. And the best example for which we have clinical data is with cytomegalovirus, where we actually have 6 mRNAs and 1 lipid nanoparticle, 5 of which have to come together to form a pentameric complex, and one of them alone, GB, is immunogenic enough to have demonstrated 50% efficacy in randomized phase 2s in the past. And so it's a very versatile platform that allows us to actually do multimeric proteins as well as single ones. Viewed from that lens, mRNA 1273, which is the SARS-CoV-2, the one against COVID-19 here on the far right, that's actually a relatively simple one for us now. It's just one chain, it's the spike protein, and we've sort of learned what a good spike protein looks like as far as conformation. I'll come back to that. This gives you a sense of where we are in the experience that we've had. So today, and I apologize, this slide is actually old, it should say 7 on the bottom. We started back in 2015 with 2 influenza, H10 and H7. We've demonstrated we reached the bar, for immunogenicity that you shoot for. It was as good or better than anything anybody had ever done with a protein subunit adjuvanted vaccine, in comparing at least across small phase 1 trials. We did an RSV trial that seemed to reach the bar of immunogenicity because you can move very quickly and change sequences here. Merck and us saw a sequence that we thought could be more potent, so that one was put in the clinic, and those results are still pending. Zika was interesting. In 2016, we started down, we took the last sequence that CDC had published without any preclinical work. We took it into the clinic. And the first one, at least at the point where we weren't dose escalating, going up to 100 microgram, we saw a smidgen of immunogenicity, but it wasn't enough. By then, we had figured out where we probably got the sequence wrong, and we had a backup that was more than 20 times, or at least 20 times, as potent in preclinical models, including NHPs. So together with BARDA, we kind of, Zika was no longer the threat that it was in 2016, at least not in the US. And so we backtracked, we put that one back into phase 1, and as we recently disclosed, we got it right this time, and a dose as low as 10 microgram is now very nicely immunogenic. Chikungunya was another complex antigen. Like Zika, it requires a viral-like particle. We also achieved the bar for immunogenicity. I've spoken about HMPV and PIV3. I'll tell you, CMV is the one that's given us the most beautiful data to date. Not only is it the most complex one to make, but actually half the population is infected and live at certain levels of antibodies. The other half is naive. We've managed to take the seronegatives and bring their immune response much higher than what it is in the seropositives. We've managed to take seropositives and even boost them further. And what this taught us, and this was unexpected, our bar was just to get to the level of seropositives. If you use this technology to focus the immune system right, you don't carry all the other baggage that a latent virus has managed to accrue over evolution in terms of its effect on the immune system, you can probably even do better. And we've completed the phase 2 of that. The phase 3 is slated to start next year. And this is the world we were in, more or less, when the COVID story hit. And so the data that we had was, we've seen over 1,500 healthy volunteers treated. We've understood the dose to probably work optimally between 25 and 100 microgram is where we see the biggest bang for the buck. We've seen neutralizing antibodies in every one of the applications and across all 8 viruses for which we've tried. And the safety profile is consistent with that of marketed adjuvanted vaccines, an even mix between local reactogenicity and systemic flu-like symptoms. Nothing really surprising or untoward. And so that brings us to SARS-CoV-2. So here, I give a lot of credit to both Stefan, my CEO, and the team that for years has been committed to leverage this technology for the benefit of a pandemic, even though for a small biotech commercial aspiration company, that's not necessarily where you go. If you look at our proof of concepts, they were to begin with against 2 pandemic threats for influenza. And we had been talking to Tony Fauci and his team in October of last year. I was down with Stefan at the NIH, and we were actually, the NIH, to give them credit, were always worried about the US not being prepared, and they have been looking at technologies that could react quickly. And so we had been talking about a demonstration project for this program. They had, together with us, we've shown data preclinically in MERS that this technology can actually give you a great neutralizing antibody. If you've seen the data on the previous slide, 5 of these viruses have pneumorespiratory flu in their name. So we're not strangers to respiratory viruses, RNA viruses at that. And so the ability to move quickly, then we were thinking about a demonstration program when in the end of December, beginning of January, it became clear that something's coming out of China. The sequences were published, and then we very rapidly moved to antigen design within a couple of days, put it in production. And because this is really synthetic enzymatic biology that happens in an aqueous phase, you don't need cell lines to scale up. You're not limited. A large scale reactor in a recombinant technology is 30,000 liter vat that requires 3 stories of a building and $1 billion in capex. For us, a large scale reactor is 30 liters. Because it's all in aqueous phase, it's an enzymatic process, and it works fast as well. So it's a matter of days and weeks, and you get the plasmid and then you put it into production. And so in 42 days, we were able to release it. Another 3 weeks went by for the final IND review and getting the clinical trial started. And so in practically a couple of months, we went from designing the sequence and putting it into production to dosing the first volunteers. Now, the critical element that allowed us to get there fast and start fast is also the critical element that is allowing us to scale up production. So a few years ago, we had already invested in having our own manufacturing facility, and that's where practically all of our GMP product is coming from. We have enabled some CMOs. We were looking ahead. As I said, CMV is slated to start its phase 3 trial next year. So we had already, we're planning what would scale up look like, what would a commercial company footprint look like. This is, of course, accelerating all of that. We're very happy to have a collaboration now with Lonza, where we're tech transferring to some of their suites. And if you kind of think of what's ahead of us, we should be able to leverage the data and that collaboration with Lonza to enable scaling up production such that we envision up to 1 billion doses, assuming a dose is 50 microgram, starting in 2021. We're already starting to produce and scale up right now so that we're ready even earlier, certainly on the US side. The last point I would make here is that this has truly been a public-private partnerships. I've done several of these in my career. Never have I had the luxury and pleasure of this level of just frank talk and collaboration that takes away all the bureaucratic hassle. In some ways, this pandemic is bringing out the best in all of us, and I'm tremendously indebted to my colleagues in the various US government branches that have actually stepped into the breach, starting with NIH, NIAID to lead the first phase 1 trial, to lead the assay development and the animal model development, and connect the academic dots in a way that allows us and everybody to come up after us to harmonize what it is we're doing. I think many of you likely have seen the harmonization phase 3 discussion that Tony Fauci and the NIAID team had published 3 days ago in Science. We're absolutely supportive, and in fact, this has been a tremendous and continues to be a tremendous enabler for us. We got a large grant from the US BARDA to scale up and accelerate development, both the clinical development and the manufacturing scale up. Our IND has been submitted and opened recently for a phase 2 trial. We've got fast track to that. That's really meant to dose confirm and give us more safety data on the road to the phase 3. And our phase 3 trial, we expect now is going to be a very strong collaboration with NIAID to launch this summer. I think we're completely in alignment with the agency and the other stakeholders of wanting to generate appropriate randomized placebo-controlled data in the context of this pandemic to be able to prove the benefit and describe the safety in a rigorous and diligent manner. And we're also cognizant of the fact that with emerging data for immunogenicity and an understanding of how those data may connect to convalescent serum and animal models, I think there's going to be a keen interest to understand what to do with those data and how to accelerate even further the phase 3 trials so that whatever usage we have in the population is fully informed with the data. I don't have data to share with you to date. Our preclinical data has not been published yet. I have personally not seen the clinical data yet. We expect to be able to speak to both of those in the coming weeks. And I think the last point I would make before opening the floor to question and answers is, people ask me, when will a vaccine be available? And my answer is twofold. First, this is not going to be in our communities in a bathtub moment. It's not going to be a black and white. I think as we get more data, as we understand the safety and efficacy, we will be ungating the ability to deploy this vaccine in a safe and diligent manner. And I foresee that to continue to be a strong collaboration between us, the US government, and other governments as we scale up to meet this goal, not just for the US population, but the rest of the world. And in that context, the partnership with Lonza is enabling us to increase our manufacturing footprint beyond just the US. The second point I'd make is that this is really going to require investment at risk and scaling up manufacturing and production. Much has been said about that, I think, in various forums. We've been doing our part here, starting sort of on our own dime and quickly with BARDA funding and hopefully soon with additional support from others to continue to deploy capital at risk with the expectation that we will generate data that would support the widespread utilization of this vaccine. Let me stop here, and happy to take questions.
Sure. Thank you very much, Tal. I guess I'm not sure if this is in scope for what you can answer, but I'd like to start a little bit with the immunology of the construct and what it is that your platform technology actually does. And you mentioned, for example, that viruses get into cells and it gets into the cells and does much what a virus does. But of course, a virus contains genes that inhibit the innate immune response and things like that. Can you tell us a little bit more about your technology and why it is that you get high expression levels of RNA from this technology in particular, and how it is that it avoids the typical problems of low RNA stability and innate immune responses after RNA finds its way into cells?
Yeah, that's a great question. I'll make 2 points here. I think it's a function of both the chemistry and the process. I used to rebel at the notion that we're regulated as a biologic because you could make all of this in completely synthetic mechanisms. But the truth is that the process here really does matter. And so if you look at our IP estate and you kind of connect the dots, and a lot of it's on our website, having the modified uridines as part of the mRNA is something that a virus can't do. And so I think one of the seminal observations has been that, TLRs inside the cell, they are primed by evolution to be the cell's fundamental way of detecting mRNA where it shouldn't. There is nothing more scary to a eukaryotic cell than foreign nucleic acids coming from the outside to take over the cell, right? And so evolution for eukaryotes has been so scared of that we put an extra membrane between our genes, the DNA in the nucleus, and the rest of the world as the cell sees it. Now, the primary mechanism from an evolutionary perspective has been to focus on that one nucleotide that's different between RNA and DNA, because that's how the cell can distinguish that it's got RNA in the wrong place. And viruses have no choice. They have to take over the cell's own machinery to make RNA, and so they're obligated to use the endogenous RNA. And I think one of the seminal things for us in terms of the potency, the ability to get a good production of RNA without tripping up that evolutionary tripwire of TLR activation in the innate immunity has to do with that. But that is just one small piece of the puzzle. The two other pieces have to do with the composition of the lipid nanoparticle, the kinetics of it, and the ability of it to deploy its cargo, and the process by which you both make these constituents and put them together. And if I would summarize what this company had done before I joined, and even more so since then, compared to my days when I was in the lab trying to make RNA, and you could make RNA in a lab and get a smidgen of it to express in a mouse, the potency of where we are today is probably at least 4 logs higher on a mg per mg basis of how much mRNA you give to how much protein you can make. So I think that's a fundamental aspect. The other element, to your point, is we don't carry the baggage. So we get the antibody. T cells are going to come to the game because they see epitopes that they don't normally see. There's probably enough innate activation going on to facilitate all that, and the proof is in the pudding. And the best example I have is, again, those CMV data. CMV is notorious for having all kinds of IL-10 mimetics and other stuff it uses to dampen the immune system. It allows itself to be latent. If you just give the antigens and you focus the immune system's attention on that without all the rest of the baggage, you can go to at least a log higher of an antibody concentration. You get very nice and very potent boost in persistence. And that's the piece where if you even take a replication deficient CMV, and our competitors have done that experiment, you can, with that, build a vaccine that will boost seronegatives to the level of seropositives. You don't go above seropositives, and you don't boost seropositives beyond their natural state. Now, to answer your question on this one is simply the spike protein. It's got 2 modifications that have been put in place to stabilize the conformation and sort of the pre-fusion state, if you will. And that's based on a lot of learning from Barney Graham's team over the years, and that's been validated in the MERS system.
Okay, and do you know for sure that spike protein is expressed as a trimer?
I don't think we've published any of that yet.
Does that mean you don't know, or does that mean that you do know?
That means that as an officer in a public company, I can't speak to stuff that are not in the public domain.
Got it. Okay. You mentioned that in your Zika vaccine, there was a big difference in immunogenicity between the 2 constructs. You attributed it just to the construct. But of course, you've selected this SARS-2 construct based on, as you mentioned, pre-fusion stabilization and other things, and the virus continues to perhaps mutate. We learn now that the European strains seem to be more prevalent than the original Chinese strains, which is the sequence that things are based on. And of course, it may change further. So how sure are you that you actually have the right sequence here?
Yeah, so I think that it's very different from the Zika story. The Zika, because it is a viral-like protein that has to get cleaved and reassembled, and it's got a leader sequence, because you're really taking just a part of the genome but you have to make sure it gets transported in the right place so that you get the right sort of production of a viral-like particle. It turns out that you can screw that up in numerous ways. We changed both the sequence of the viral itself based on our understanding of what the right sequence was. We also changed the leader sequence that's meant to, we took one from sort of a different construct that Mike Diamond had showed us better. So there were several things I think that went into that optimization that we were blind to at first. I think the spike protein is a much simpler story in the sense that it's just a membrane protein, and what you see is what you get. And we've done that numerous times without a hitch for the other respiratory vaccines, whether it's HA antigens into flu or your F protein for RSV, HMPV, and PIV3. So in my mind, it lives in that world. Now, how sure are we in terms of the actual sequence? I'll give you again two answers. One is that once we see the ability to prevent disease in animals and protect from challenge studies, and we see neutralizing antibodies, then I think we would be reassured that we have the right immunogen. And I think the other part of the story is we expect and intend to prove that indeed that immunogenicity can prevent disease. That's what the phase 3 trials are all about. I think what it will do is also establish a new platform concept similar to flu, such that if this thing mutates to the point that prior immunogenicity is not good enough, well, our platform can change as fast. You just give me the new sequence and off it goes. And once we would have demonstrated that in principle against a circulating strain, you've got an effective vaccination platform, just like we do with the flu. Every year you get the sequence, you put it into production. Nobody questions whether it's going to work. Everybody accepts that if it doesn't, it's usually because by the time you put something in production, you actually, you mischaracterize the strain. And I think the last element is this is still more stable than flu because the beta coronaviruses have a proofreading enzyme. And so I think all that's been described to date is still not anticipated to materially change the immunogenicity and the immune response against the spike protein. But of course, we're going to have to prove that.
Got it. A number of questions still about your platform, and can you say a little bit more about how your platform differs from other competitive RNA vaccine platforms, or for that matter, DNA vaccine platforms, and not just in how it looks, but also, do you know anything about comparative type of immune responses, in particular T-cell responses against one of these kinds of platforms versus another, at least based on what's been published?
So let's start with some of the fundamentals. I think against other RNA platforms that are not using the same chemistry, I think they're going to run into challenges in terms of how much protein they make. You could make the argument that non-specific activation of the innate immune system will compensate for your less efficient protein production in the context of a vaccine. That may be okay. That may be the case. I don't know. The truth is that the companies that are saying that and doing that actually have very little clinical data on infectious disease vaccines, if you look carefully. And none of them have anywhere near the database, the diverse database of phase 1 and soon phase 2 trials with so many different infectious disease vaccines across so many subjects treated. So I think our clinical understanding of the performance of our platform is at a much more mature level. Some of our competitors are still trying to figure out what is the optimal chemistry, what is the optimal design for an antigen, and what is the optimal, everything else. It's still in phase 1. I think we're well beyond that. In terms of the other question you asked about the T cells, BioNTech specifically, I think, started there and has most of their experience in T cell applications as they relate to cancer. I think everywhere we've looked, we've seen commensurate T cell responses. You would expect it based on the fundamentals of biology, which is you get the endogenous protein production from within. that application for us, we've demonstrated the ability to do T cells there. When we've looked in our clinical trials, we have seen T cells. We've recently spoken, I think, for a couple of those vaccines about those data. I think interpreting those data in clinical human studies is super challenging because you're at the mercy of very tiny populations and the time at which you get, and they're very sensitive assays, so the variability tends to be very high, and they're nowhere near as tight and clean and interpretable as antibodies. But we've consistently seen T cells. We've consistently seen, and I think this is also worth mentioning in the context of COVID, we've consistently seen the right kind of T cell responses. So people kvetch about, enhanced disease and is this going to be the formalin inactivated RSV of the 1960s? And when we do it in those comparative trials where you have a positive control, mRNA technology in our hands with what we've shown with the antigens as we've designed them has never shown us that kind of a wrong directional priming towards a Th2 or enhanced pathology. And so I think we're pretty confident on that front, both from the T cells that we do see and the phenotype of those T cells and the rest of the antibody response that we see, that we are generating the right kind of immune response. I can't speak to where others are in terms of process and experience. I think a lot of that sort of lives within every company's know-how, but I will say that we have benefited tremendously from the acceleration of our prior experience in the clinic and infectious disease, the building of our own manufacturing facility several years ago, and the close proximity between our research platform and the technical development and the GMP manufacturing, so that I think our understanding of the performance of those aspects, which are critically important, I think is quite mature relative to our age and relative to probably some of where the competitors are.
Thank you. So a lot of questions about results, perhaps not surprisingly, because other speakers in the series have presented immunogenicity results in animals, and in some cases, of course, before moving forward, one would like to see some protection in animals and would also like to see the immune responses in humans, and at least perhaps try to compare those immune responses in humans to what is seen in convalescent serum from people who've recovered, to get a sense of whether these immune responses are high enough. And you mentioned that you didn't have any of that data, but I couldn't help notice on actually this very slide that's up, slide 8, that you, of course, started your phase 1 trial in April, but you're about to start your phase 2 trial in the second quarter, and you're proposing to go into phase 3 by the third quarter, which implies that there must be some data that is supporting this very rapid timeline. So can you tell us a little bit about any data that is supporting the timeline, or if you're not allowed to talk about it now, when those data will be available for people to look at?
Yes, Phil, thank you for the question. I would just say that I expect the data to be available for us to be able to talk about it and share it in a cogent manner in the coming weeks. And I think that pertains both to the preclinical basis of expectation of benefit as well as emerging clinical results. I think when we have them in a way that we can talk to them in a way that's cogent and understood, then we will. And I would just say that everything we've seen so far supports our belief that these timelines are achievable, and we're working towards them. And I would say that we're doing that in the only other thing I'd say along those lines is this continues to be a very strong collaboration with NIAID. They are running the phase 1 trial. They're at the forefront of developing the clinical assays for all of us, and I'm immensely grateful, not just on behalf of Moderna, but on behalf of everybody else. And I think that, as that data emerges, the ability to react to it quickly between us, NIAID, and FDA is also enabling our anticipated timeline.
Thank you. What can you tell us about how scale up of the vaccine will align with access? It's encouraging that you'll be able to ultimately make up to 1 billion doses per year. Of course, there will be substantial worldwide demand for vaccine, And so there are some questions about how the vaccine will be distributed. Where are the primary markets that you're going to be looking at?
It's a great question. I would say the following. First, I think we need to distinguish between the ability to manufacture in bulk, the ability then to fill, finish, and distribute, and finally, who gets it, when, and why. I think the ability to manufacture in bulk, we're very confident in our capabilities and capacities as we've described them. We've spent a lot of time already understanding this platform and its ability to scale up as we were thinking about CMV, which, as noted, is actually a much more complicated one because it has 6 mRNAs that need to go into every vial. So relatively speaking, I think we were ahead of the curve. And so we're leveraging all that know-how and now the collaboration with Lonza to increase our manufacturing footprint. I think the thing that everybody is going to be challenged with in the coming months and years is going to be sort of fill, finish, and distribution. We're actively looking at that, but all of this requires significant capital to be deployed at risk. I think that's where, for us, having a strong public-private partnership, both in the US and ex-US, is going to be critical. It's either that or find other sources of capital. The reality is that that's where some of the big pharma companies may be better positioned, and we're well attuned to that, and I think that's fine. The world needs all the vaccine it can get. The last point I would make has to do with distribution. I think I would make the distinction between the pandemic need and the sort of what happens next. I think the first batches that could be available in any geography and get licensed, or whether it's an accelerated approval or an emergency use or treatment INDs or what have you, there's going to be an urgency to start with vaccinating those that need it the most. In the context of a private-public partnership and in the context of this high unmet need, my expectation is that we would work with the US government and other governments to figure that out. I don't think we, or any pharma company for that matter, are going to be appropriately set up to determine in any given population who should get it first and how. So I think those discussions are already happening within the various branches of the US government, and we look forward as the data matures and our production matures to having a line of sight to what that looks like. I think to the degree that this becomes sort of a multi-season thing that lives with humanity for a long time and maybe requires sort of more, it becomes more of a flu story than it does, a 1918 flu story. I think that would then change over time to a different distribution model, but I think it's too early to speculate on whether that would happen and what that would look like. And certainly we're focused on the pandemic need right now.
Thanks, Cal. As a 2-dose vaccine, of course, in a pandemic, there's always the question of how long it will take to arrive at full immunity, or whether there's sufficient immunity after a first dose to make a difference right from the get-go. Can you say anything, perhaps from, well, whatever you know about this vaccine or other related vaccines, mRNA vaccines, about the evolution of the immune response, as well as the dosing interval and the time at which, after vaccination, after initiating a series, you would expect to see the optimal immune response?
Yeah, a great question. So let's step back to the basics here. I think that biology and immunology has taught us that a prime and a boost is superior, I think, in every case that somebody is naive. Now, you may sometimes get a prime that is good enough, and therefore you don't need the boost. But from an evolutionary perspective, you give a prime, it lasts for a few days. If you come back a month later and wake up the immune system and say, hey, no, this thing you thought you cleared, you didn't, boy, that's scary from an evolutionary perspective, and our body reacts appropriately, and that anamnestic response now is faster and much more vigorous. And so, of course, we'll be walking around then with a higher titer of antibodies. I think we need to make a distinction between the fundamental immunology and then our expectations from any given platform. So there's a lot of talk about the adenovectors needing only one shot. Well, the truth is that the adenovectors can't boost because the immunity you get from the get-go is so strong that you've generated now an immunity against all the other elements of that. In fact, this is the same technology that's been adapted from the world of gene therapy, and we all know that in gene therapy it's a one and done deal, not just because you need it from the DNA perspective, but often because the vector doesn't allow you to go back in a second time. So it's conceivable that this technology can work after a prime to sufficient level that you wouldn't need a boost. But I expect, based on first principles and the experience we've had where we have prime boost, like CMV, is that while a prime looks very strong, the boost actually adds on more. And so in the context of a pandemic, you really want to get the most robust you can. So it'll be terrific if we show that a prime was enough to already decrease cases, but a boost added additional benefit, because we're trying to prevent everything we can in the context of trying to get the population immunized, not just disease prevention, but also prevention of infection and transmission.
Thank you. A number of questions still about the vaccine, in particular the role of the LNP, which you mentioned as being an important part of the vaccine. Is that primarily playing a role in bringing the RNA into the cells, protecting the RNA, or is it also by itself influencing the immune response? What can you tell us about that part of your technology?
Yeah, so our understanding of the LNP is that it's fairly innocuous, and its primary function is to prevent the RNA from being degraded and get it in the lymphatic system to the local, to the lymph node. I think we had a lot of discussion in the early days. Well, is this adjuvanted? Is it not adjuvanted? It's not adjuvanted in the classic sense, certainly. And I think there's alignment with regulatory authorities that is considered non-adjuvant from that perspective.
Thanks. Is there anything else you would like to say about the overall immune responses with your platform versus other platforms? I think that obviously it's a fairly new technology. None of the vaccines you've put forward have made it to phase 3 yet, at least as you presented it. And yet, of course, this one is moving forward very rapidly and maybe in a phase 3 trial very quickly. So what is your level of confidence that the immune response really will be adequate to protect?
So, you know, if I've learned anything in my experience in drug development, is that you're much more likely to succeed if you follow the science and you don't take any shortcuts and you've got the right building blocks. And I think we've done that now, not just on individual applications, but on the platform as a whole. You know, we have tried to generate immunity as measured by neutralizing antibodies against 8 different viruses. We have succeeded every time. We've described a safety profile that's consistent with that of other marketed adjuvanted vaccines. And the fundamental biology here has been worked out both on first principles of generating antibodies and T cells, and a very rich body of data. I mean, I'm a clinician, so I go to the clinical data, but all of this stands on a mountain of preclinical data done with the best academic labs across many different types of viruses in many different non-clinical species. So yes, we haven't taken anything yet to full licensure. Our clinical data have only started, as I said, at the end of 2015, our clinical experience. But I think within that time frame, we've connected the dots and are moving it in a way that I feel fairly confident. I don't know what I don't know. It's true that we've not demonstrated that we prevent disease, but we are leveraging sort of the scientific know-how of understanding the relevance of an immune response as measured by both T cells and antibody levels to predict for that clinical outcome. Over.
Fantastic, Tel. And one final question. Are you thinking of this as a vaccine that might ultimately cover multiple different pathogens? Obviously, if there were evolution of the coronavirus to new strains. One could conceivably, I suppose, include RNAs for multiple strains, or do you think that doing that would I guess change the doses and would be more difficult to do, or is that something you've been thinking about?
Yeah, absolutely. I think this platform challenges our conception of how to do it, but it is absolutely feasible. We've already demonstrated that in the combination of HMPV and PIV3 that you can do that. You should expect to do that, and I would envision down the road the ability to combine numerous mRNAs. I mean, CMV is 6, 5 have to form the same antigen, but you get 2 separate antigens in that vaccine. You can measure their effect separately because they neutralize different activities, the antibodies that each generate. So I think that's absolutely in the future, but I think today I've got 2 very big competitors, the virus and the clock, and we're doing everything to beat the virus using the biology of our platform and to beat the clock using the ability of this technology to scale up quickly.
Sounds good. And I'm sorry, one final safety question. You mentioned, of course, that the technology seems to give rise to a Th1 biased response, as well as it looks like good levels of neutralizing antibodies, both of which are outcomes that one would hope would reduce the likelihood of enhanced disease. And yet, of course, the press has mentioned enhanced disease as something that's important, and there is a history in SARS and MERS vaccines in animal models of enhanced disease that resembled that associated with RSV. So can you say anything more about enhanced disease, including any other studies that you're planning to make sure that enhanced disease isn't an issue with your vaccine or your platform?
Yes, let me make 3 points and then I'll close. I think my understanding of enhanced disease is that it is a function of a heterologous prime boost or an antigen that is misfolded or is chemically, modified the wrong way. I think that both of these are very unlikely with our technology. That's number one. Number two, the preclinical data that we are generating and have been generating together with NIAID all support that this is a very low risk. It's never going to be zero until we run large phase 3 trials, but the preclinical data so far are looking supportive that this is not a big deal here. And the final point is, I'd say enhanced disease, by definition, there's no case definition for enhanced disease. It's an endpoint that you collect when you look at disease. And so the only way to put this to bed is to go run a proper development program, have placebo-controlled trials, and demonstrate that your case rate is going in the right way and not the wrong way. And we'll do that with all the typical, unblinded DSMB type of monitoring to reassure ourselves and the public that this is just a theoretical concern that has no bearing on the ultimate effectiveness and safety of this vaccine.
Thanks a lot, Tal. We really appreciate you staying even a little bit longer than you had originally planned to. So I think we're going to close it here. Greatly appreciate the clear presentation of Moderna's plans, and of course look forward to seeing some of the data that will be coming in the upcoming weeks. And of course, we all wish you luck. All of us want a vaccine that will be safe and effective. And it sounds like you've gotten to the scalable part as well. So it sounds like there's a chance that this could be one of the vaccines we're all looking for. So thank you again, Tal. Greatly appreciate your being here.
Thank you, Phil, and thank the audience. And apologies, I have to run. Have a great day.
Okay, Wing, I don't know if you'd like to say any final words to the group.
Yes, sorry, I forgot to unmute myself there. Thank you all of our speakers this week, and of course to you as well, Phil. Lots of compliments there, being such a fabulous moderator. It's not an easy job to try to navigate through all these questions. So thank you also to our active listeners for all of your questions. We've done our best to try to address those as many as we can. So just a final shout out to our webinar sponsor as well, TriLink Biotechnologies. Appreciate your support. And if you missed my introduction at the beginning, this session is video recorded. A few of you have asked, and you will get a link to it in 24 hours. Just a final note from me. Hopefully you can all tune in again the week of the 15th of June for our third series, more details will be posted on the website shortly, so do stay tuned. But in the meantime, everyone, thank you so much again, and stay safe, speak soon, and goodbye. Thank you.
Bye-bye.
Thank you. Goodbye.